Good afternoon, everyone. I'm Sewell. I'm one of the residents.
I'm Matt Kornswiet, also one of the residents.
On our developmental block. So today's Noon Conference is about some genetic abnormalities that we can commonly see in the developmental office. And we kind of did a quick overview of quite a few different ones just to kind of help us piece together because there's so many. So we did some of the more common ones and kind of tied it in with other genetics lectures that we've had recently. So did a little less emphasis on those ones that we covered.
So Dr. Cohen's going to be doing an overview to start, and then Dr. Krapf at the end. And then once we finish up, we have some board review questions at the end, hopefully if we have enough time for them. Enjoy.
All right.
So I'm going to have you pull me off the podium when my time is up. My name is Bruce Cohen. You may ask, why is a neurologist talking about genetics? It's estimated that 40% of the human genome is exclusively brain, spinal cord, and muscle specific. And another 20% of the genome, so a total of 60% of the genome, has something directly to do with brain, spinal cord, or muscle function. So as neurologists, we own the genome. I mean, we're the number one voting block of the genome. So that's the first reason. The second reason I think I'm up here talking today is my credentials have gotten me to the point where for 6 years in a row, I wrote the board questions for neurology recertification examination. And so the rules that govern how the American Board of Psychiatry and Neurology give tests are exactly the same as the rules that govern how the American Board of Pediatrics writes their exams. And it ends up, it's not so easy to write an exam question. It's got to have a very short and specific stem, and the 4 answers have to be mutually exclusive and crystal clear as to what the right answer is and evidence-based. So when you take all those rules, there's very few ways to ask a question on sickle cell anemia. There's very few ways to ask a question on any one disease. So, as an inquiring mind, you want to know everything about every single topic. But when it comes time to taking an examination or condensing knowledge, there's the ways to look at it end up being very few and far between. So I've given this lecture in various forms to pediatric groups, including board review courses, for over 20 years. And the feedback I've gotten when I've given it in the CME format is, thanks, the information you covered was in fact on the board exam. So maybe give you some background of why we're doing what we're doing. So the information in the handouts and the information you'll hear in the second part of the talk is based on the common, by the way, I take pediatric board exams, so I'm actually a consumer of the product that you will be taking. I stopped taking them this last cycle because I just don't need 2 boards, but I had taken them for several generations. And so I got to see the questions from the consumer point of view. So I'm going to step off that a little bit and talk to you a little bit about genetics, because it ends up being a very, very interesting topic to me, and will be a very interesting topic to you if you're not interested in it yet. In the next 5 to 10 years, you're not going to be able to open up a chart on a patient with any illness without having to, come up face up against genetic testing and where we're going with that. So here are some of the topics going to be talked about today, but we're going to step back and talk about the human genome and some fun facts. So everyone would say, well, gee, what's the ultimate storage device? And you can go to any drugstore and buy an 8 or 16 gigabyte thumb drive that are about this big. But in fact, the human genome is even a denser storage device, and there are people in the IT world that are talking about storing data in the nucleus of cells, because you could actually, it's a denser storage, and every bit is accurate. So first of all, if you stretched out, if you took a single cell out of your body, like a white blood cell, and then you took the DNA out of that cell and stretched it out, it would be 6 feet long. Okay, and you're shoving it all into a 20 micron diameter, cell or nucleus. So it's coiled up into this very small space, and when you compare what's in a human genome, a single cell, compared to what's on a thumb drive, it's about the same. So in a human genome, there's 6.6 billion bases, which corresponds to about 8 billion bases on a thumb drive. So, that's just unbelievable in terms of its density. As it ends up, we work in a mathematical base 4 when you're talking about the human genome. You're talking about 4 molecules, adenine, thymine, guanine, and cytosine, that are abbreviated by the letters A, T, C, and G. And the entire code is made up of these 4 molecules. As it ends up, the code is stored 3 digits at a time. So an ATG or an ATC or a CAG or a CCG, the way that this code is read and turned from a genetic code into eventually a protein is by reading 3 frames at a time. And these frames start from, they've been labeled. In the human genome has been labeled from starting point 1 all the way to 3.3 billion. So these 3 digits, which are referred to as the codon, correspond to one of about 21, 22 different amino acids. They're arranged in 23 pairs of chromosomes that we call chromosomes 1 through 22, and then the sex chromosomes X and Y. Why is chromosome 1? Because it's the biggest. Chromosome 2 is the second biggest. That's why they were named the way they were named. As it ends up, the 20,000, 21,000 or so genes in our genome make up only about 1.5, it's debatable, 1.5, 2% of the whole human genome, which means that 98 to 98.5% of the genome is made up of non-coding regions. These are large stretches of genetic material, the ATCs and Gs, where there are no genes in it. It's not a black hole wasteland. The role of these large stretches has obvious importance, the importance of which is being discovered little piece by piece. And there was an interesting article in the Science Times last Tuesday about one of the roles of these large stretches of the human genome. If you look at our genes and compare them with the chimpanzee's genes, they're about identical. The difference is that we have 30% more of this uncharted wasteland territory in our genome. We refer to the genetic component that are turned into genes as the exome, and we refer to the whole package as the genome. Here's the punchline. For the vast majority of diseases that we're going to be talking about, and again, there are notable exceptions, but for the vast majority, there are 2 major types of mutations. Type 1 is called point mutation, type 2 is called deletion or duplication, also known as copy number variant. Here's the difference. A point mutation is a single change in a single letter of the genome. So somewhere in this vast array, a letter that's supposed to be a T is turned into an A. And it's this type of mutation that leads to diseases like sickle cell anemia. Deletions or duplications, also known as copy number variants, are when generally large chunks of DNA are either missing or duplicated. Sort of the quintessential duplication disorder would be trisomy 21 or Down syndrome, where an entire chromosome copy is duplicated. You have 3 copies instead of 2. A traditional deletion would be something like Turner syndrome, where one of the X chromosomes in a female is gone missing. In terms of deletion, you can have a deletion of a single point. You would say, well, isn't that a point mutation? Well, not really. It's a deletion because it's missing and it shifts everything over one spot. You could have 2 nucleotides missing. You could have 3. You can have 4. Typically, you'll have 50,000, 100,000, 2 million points missing. The quintessential disorder where you have a couple million points missing would be something like velocardiofacial syndrome. And the deletion in velocardiofacial syndrome or DiGeorge syndrome is typically missing about 30 different genes. So a whole big chunk of genetic material gone. In any of our genetic materials, we are loaded with point mutations, deletions, and duplications. How does that make sense? Well, what differs me from any other male in the audience are thousands and thousands of differences in our genetic code. We're enough alike, our genes are arranged enough alike, those dead spaces between the genes are enough alike that we're all human. But in terms of our physiology and the diseases that we'll get, there's thousands and thousands of differences. These are in fact mutations off of a standard human genome. We're all different, but they're not considered pathogenic mutations. Mutation, from a biologist point of view, mutations aren't bad or good. They don't have any moral to them. Mutations can actually serve, in some circumstances, a beneficial role, helping one adapt to their environment better. And over the course of time, human populations that have migrated from one part of the earth to the other part of the earth have mutations that actually survive and are passed on because reproductive fitness is improved. So again, mutations aren't bad or good, but some mutations are cause disease, and those are the ones that we're really talking about. Does anyone have any questions so far? So this is probably the best source of material if you run into a patient on the floor that has a named disease. You just type in the Google search engine gene reviews. It'll take you to this website, and you can just type in the name of the disease. Alper's disease, Cohen syndrome, not me. But there's 638 chapters, and in these chapters there's actually some chapters cover dozens of different diseases. So if you type in spinal cerebellar atrophy, there's a chapter that covers the 84 unique spinal cerebellar atrophies, with pretty much everything you want to learn about any of them in a standardized format that's easy to digest. So here was, I typed in Rett syndrome, and Rett syndrome links you to what the diseases are now called MECP2 related disorders. Written by John Christodoulou, who's a physician in Australia who's been, he's the world's expert in this disorder. Here's one on Prader-Willi syndrome. So let's move on to what the genome looks like. Again, you got this in high school biology. I didn't get this till college biology because they didn't know about the genome when I was in high school. That's a joke. They had just discovered it. So again, this is what DNA looks like. The DNA strand is double loop. Why are we double-stranded? It helps with stability. Double-stranded DNA will sit around thousands, 10,000 in the Jurassic Park. It really does sit around for eons. RNA, a single-stranded molecule, sits around for a couple seconds before it starts breaking apart. One loop is what's called the sense strand. That contains the nucleotides that are read by the ribosome and translated into the protein. The other strand is the stability strand called the antisense. Some people refer to it as junk. It's only there for the stability. As it ends up, it probably has more of a role than we had thought. But for all intents and purposes, only one strand is read. The idea is first through transcription, the DNA is turned into RNA, and then through translation, the RNA is turned into protein. And I'm just not going to go through this process because most of you have that ethereal memory. It's out there. Yeah, I do remember that sort of. That's all you need to know, is that it's turned into protein. And then the proteins are turned into, molecules in our body that actually do something. So the insulin is a protein molecule that has, dozens of different, reactions within the body that allows it to do what it does. Thyroid hormone is another protein molecule. And all these protein molecules look differently. The backbone of a protein are the amino acids, and of course they can be glycosylated and modified on the Golgi apparatus later on in their formation. But the backbone is amino acids. And why, if you have a mutation, you would have an amino acid. If you have a mutation, it's going to cause a change in the amino acid that gets inserted into the protein. And certain amino acids look much different than other amino acids. If an amino acid looks differently, it's going to function differently, and the protein is going to fold differently and not behave properly. So here's the genetic code. So again, if the translation machinery comes across a C, that a U, and a G, it's going to be read as a leucine. If it comes across as a C, and a G, it's going to be read as a proline. Okay, if there is a U to C mutation right at this point, what was to be a leucine amino acid inserted into the protein is going to be a proline. And that can actually cause disruption in the ultimate function of that protein, and therefore disease. And then the most critical mutation of all is this UGA. When the ribosome comes across a U, then a G, then an A, it stops. That's when the protein ends and it gets chopped. If there's anything that causes a mutation, a UGC to a UGA, instead of the cysteine being inserted, it'll cut the molecule short. And again, a shortened protein molecule doesn't function. So we expanded on the two main. I'm going to expand on the two main types of mutations that occur. The first being the point mutation, and then these can be further delineated. And missense mutation is substitution of one amino acid for another. Depending on that amino acid substitution, you may actually not get that big of a deal in your protein change. So alanine and valine are two similar appearing amino acids. And if an amino acid changes from an alanine to a valine or a valine to an alanine, most often it's not going to change any function of the protein, except if it's in a real critical area of the protein. And you go along life with, let's say, insulin, instead of functioning 100% normal, maybe that insulin protein may be functioning at 98% normal, and you're never going to develop clinical disease. But if you have an amino acid substitution that, for example, that occurs in a critical part of the hemoglobin molecule that leads to sickle cell disease, well, that's a big deal. But those are examples of missense mutations. Nonsense mutations are the insertion of a stop codon where you don't want it, and those are disastrous for protein function. So if you have a nonsense mutation, instead of your protein being this long, your protein's going to be that long. The body views proteins that are this long as being foreign proteins, and it chops them up and chews them up and gets rid of it. So for nonsense mutations tend to be deleterious almost all the time. The only exception to that would be if you have a nonsense mutation at the terminal end of the protein, and so the body's going to view, instead of this protein, it'll see this protein, and it's not going to necessarily chop it up, and there may be enough residual function of that protein to do what it's supposed to do. You have the deletion duplications. Again, deletion is missing 1 to 20 million base pairs. Bases. A duplication is an addition of 1 to 20 million bases. And then there are other types of mutations. Again, we're not going to really talk much about it. I mean, the repeat expansion is a huge new area, but it's relatively few diseases. It's not that it's unimportant, but it really goes beyond the scope of this talk. The critical exception to that being Fragile X syndrome, which is a repeat expansion of disease. And we're not going to talk about methylation defects. It gets far too complicated for this disease, or imprinting, other than one of the disease that's imprinted really in Angelman syndrome. So here's an example of a missense mutation. So this is here represents what is a healthy nucleotide sequence. as it's read on the ribosomes, creating this healthy string of histidines. And at this point, you have a mutation that turns that normal A to a C, and a protein is inserted. So that's an example of a missense mutation. And again, depending what amino acids inserted into the protein and where it is in the protein, you could get no disease, mild disease, or very severe disease. Mutations probably occur. I mean, if you think about what real critical proteins are in the body, one of the most critical proteins is insulin. There's, hemoglobin's obviously a critical protein. As it ends up, sickle cell anemia is a survivable critical protein mutation. There's probably mutations in the hemoglobin molecule gene that have popped up over time that are so deleterious it's not even compatible with life, and so those get washed out very quickly, because if you can't reproduce, you're not going to pass along the mutation. Here's an example of a nonsense mutation where the nucleotide change results in a TAG, the stop codon, inserted into the protein, and then you can see the protein is only half formed. An insertion mutation, so this A is inserted into this normal nucleotide sequence right here, and it causes what's called a frame shift. So all the nucleotide sequence keeps getting read, that every single amino acid afterwards is going to be an incorrect amino acid. And those are disaster situations as well. And again, you can, instead of adding an amino acid, a nucleotide, you can subtract a nucleotide, again, causing a frame shift in the reading frame, leading to every amino acid added to the protein is incorrect. You can have duplications on a large sense. So this is an example where a segment of a chromosome is duplicated, and you could say, well, is too much of a good thing bad? Trisomy 21 is an example of that. You have an extra copy of chromosome 21, and yeah, that's bad, because you can have a gain of function disease associated problems. Again, this is another example of a frame shift mutation. So this is a colorized photograph of human karyotype, and we're going to talk a little bit about how these are done. So this is, again, another karyotype of trisomy 20 of a child with trisomy 21, showing an extra copy of the 21st chromosome. This is a patient with Klinefelter syndrome, showing 2 copies of the X chromosome and a single copy of the Y chromosome. And again, this is a child with Turner syndrome, showing a missing Y chromosome in a phenotypic female. So how is karyotyping done? You basically take peripheral blood, you extract, you create a lymphocyte line, you add a mitotic blocker like colchicine, and then you bust apart the DNA, and what you end up getting is the DNA appearing as this little pretty picture. And then someone actually takes a photograph of this, and this is what the photograph may look like. And then you actually need a good pair of eyes and scissors to cut it up. So the way karyotypes had been done historically is off a photograph. This is not really a high quality photograph, but this is how the old karyotypes looked. And then you'd actually have to pick off, oh, this is chromosome 1, this is chromosome 2, and then these would be stained. And the 550 bands is sort of a typical staining of these, and what 550 bands means is that when you get a good pair of eyes, you can actually, after reading all of these, you come up with 550 bands. And a cytogeneticist, who are sometimes physicians and sometimes PhDs and sometimes technicians, could look at this and say, there's no missing parts. no missing parts to the resolution that you can actually see these 550 bands. And so this, when you get below, and some people say 1 million missing bases or 1 million addition bases, this is the limitation is that you could be, missing 500,000 bases and you're not going to see anything abnormal on a karyotype. So this ends up getting us part way to understanding childhood onset human disease. So back when I was at your education, the stage of education, this is what I had available to me for genetic testing. This was it. And so we were able to diagnose about 200 different named diseases, which I've talked about today: Turner syndrome, Klinefelter syndrome, trisomy 21. Most of the large trisomies are the monosomies. Were able to be diagnosed this way. If you took a disease like velocardiofacial syndrome, where you're missing 30 genes, you get a normal karyotype of 30 genes. So this gets you so far. So if you take a child with intellectual disability, and that's all you know about the child, you don't know anything about the family history, you don't know anything about anything except intellectual disability, there's about between a 3 and 5% chance you'll pick something up on karyotype. So we talked about what these limitations are. So karyotype is not going to pick up a point mutation. It's not going to pick up a small deletion. It will probably pick up a deletion under 2 million base pairs reliably these days. We're still doing karyotypes. We still offer them in this lab. Some of the scientists that have made their careers on karyotyping are now at the age that they're retiring and have come out and said this is a missing, this is an art, a dying art that will disappear with this generation. And very much like good artists, if you go into an old house and look at the artwork, the woodwork in the house, you can't duplicate that today. So karyotyping probably will never be completely on its way out, but the high-level karyotype scientists are sort of disappearing. I remember learning about this technique when I was in medical school, FISH. It was a laboratory technique. It wasn't for clinical use. It stands for fluorescence in situ hybridization. And it's based on the fact that you can develop a probe. Now, how people develop these probes is by, a lot of it's by accident, a lot of it's doing a lot of hunting. The original FISH probes people would work on for their PhD thesis to develop a probe. And a probe is basically the oligo target to a stretch of DNA that you know what that stretch looks like. So again, back in the late 1970s, this was a developing these probes again took months and months or years to develop a probe. And so you put a fluorescent label on the probe and you can say, let's go looking for these 30 genes that are missing in velocardiofacial syndrome. And you'd have a probe and you'd look for it, and instead of seeing it in a healthy person, normal, genetically normal person, you would see 2 probes light up because they're 2 copies of that stretch of genetic material. And someone who is missing that stretch of genetic material, the probe would attach normally to the stretch of genetic material that was there, but in velocardiofacial syndrome, you're missing a chunk of DNA, and it would not attach, and so you would see a single fluorescent pattern light up. And so that was the original FISH in situ hybridization assay. Then someone said, well, what we ought to do is have a way to really control for this, and the way to control is to find a probe for a stretch of DNA right next to where the missing stretch was. So that's what this concept is. So if the pink is for the region that's missing in velocardiofacial syndrome, the green was a probe designed for that stretch of DNA right next to it. So again, in a normal control, you'd have a normal green and pink signal on 2 parts of chromosome 22. And In a person who's actually has deletion, you'd have the control and the area in question light up on one, and just the control light up on the other. So, the thing about FISH is that it's highly accurate. It's highly accurate today. The bad thing is you have to know what you're looking for. So you would have to say, I think this kid has velocardiofacial syndrome, and let's order a FISH assay for velocardiofacial syndrome. Or I think this kid has Williams syndrome. Williams is not the best example, but let's order a probe for Williams syndrome. And each probe was a different test, and each test costs a lot of money. And we actually developed this we say, we're going to need to go fishing on this kid. Because we knew the kid had something, we just didn't know what it was. And sometimes you'd go fishing with a lot of knowledge, and sometimes you go fishing because you just didn't know what the child had. And so, again, these FISH assays became online, clinically available in the early 1980s. And they were very helpful in diagnosing a few more percent of kids. So again, the limitations to the FISH assay is you have to know the disease that you're looking for to order the right test. The second limitation is that you're looking for DiGeorge syndrome. It's only good for DiGeorge syndrome. And by the end of the day, there were about 200 different FISH probes designed for human illness. And the other complicating factor is if you wanted a FISH probe for velocardiofacial syndrome, there were probably 50 labs around the country that could do it. If you were looking for one of the rarer diseases, you'd actually have to find the laboratory that was doing it and send your sample to that laboratory. And try to do that in the days before the internet. I mean, if I asked you any question, the first thing you do is pull out your cell phone. You'll have an answer right away. So it wasn't so easy. So someone said, let's put all these fishes together. How am I doing? How much time do you need?
5 more minutes.
5 more minutes. What if we put all these fishes together and we microsized it? Well, that's what someone did. So this started happening in the late 1990s and early 2000s. Someone took a glass microscope slide and drilled a couple hundred holes in that slide, not all, a couple hundred wells into that slide. And they basically said, we're going to put velocardiofacial here, and this fish here, and this fish here. So they lined this up with all the 200 fishes that they had, and they said, where else are human deletions and duplications common? And it ends up where the two parts cross together, the centromere, lots of deletions and duplications there. And so they randomly said, we're going to develop FISH probes in the middle of these highly dense regions and just go fishing for diseases we don't even know about. So this actually became known as the subtelomeric FISH test. And it was the first iteration of the chromosomal microarray. And I believe the first time I actually ordered one was 2001. So by 2005, there were over 1,000 probes available, and so these were still being done on glass slides that were being transitioned over to computer chips. And you can actually see stretches of DNA with 2 copies and then a 3rd copy or a missing copy. So this is what the printouts would be. And that would signify, I don't know if that's gain or loss of function, I don't know if that's a duplication or deletion, but you would see the signals duplicated or deleted. And the SNP arrays that we use now, again, these have gotten more dense. There's over 2 million probes currently in the current SNP array. So the current chromosomal microarray you order now is over, I think it's about 2.5 billion probes. And it ends up being an incredibly robust test for patients with intellectual disability. And it is now part of your guidelines, American Academy of Pediatrics guidelines, American Academy of Neurology, that if a child has intellectual disability, it is reasonable to order the chromosomal microarray test for investigation. The hit rate's about 7%. So I think this is incredibly important for pediatricians to know. because this is available to you in certain parts of the state of Ohio and around the country. This test is not marketed to neurologists. It's marketed to the general pediatricians. You're sort of protected from that in the Akron children's world because of different policies and that are in place, and because of a relatively strong genetics department that is controlling some of this. But again, if you go out into the true private practice world, these folks are knocking at your door every bit as much as the amoxicillin salespeople are trying to get your business for this test, which we're going to be running in-house at the cost of about $450. Okay, what does an MRI cost? 10 times as much. Just think about it when you're ordering a test for intellectual disability. The marketplace, these tests run up to $7,000. It's the same 2.5 billion, probe assay. So, again, just to keep that in mind. So, one of the things that I'm proudest about that I worked on was a template policy that has been adopted by a bunch of insurance companies for chromosomal microarray analysis for intellectual disabilities. So, I'm going to stop here and let you do your thing.
All right.
Thanks.
So I'm Dr. Krapf. I'm one of the neuropsychologists in neurobehavioral health. And as we were talking about preparing this lecture, one of the things that Dr. Cohen mentioned is that as you're studying, particularly over some of these more rare diagnoses, that really all you need to know is probably 5 bits of information. And if you have 5 bits of information about a diagnosis, you're likely going to be able to recognize some component of the question that's on your boards. So I cheated a little bit as I was making these slides because it's hard to boil down diagnoses to only 5 pieces. But hopefully these will be kind of things that stand out and help you answer questions related to these different diagnoses. So we're going to do kind of a whirlwind tour of some of these and then get to the board questions. So the first one we're going to talk about is Klinefelter syndrome, which is XXY. So these are male patients that have 2 or more X chromosomes. And this affects about 1 in 500 to 1,000 males. It's suspected that this diagnosis is underdiagnosed because mild cases may not have significant enough symptoms to warrant treatment or warrant investigation. Characteristics include small testes, insufficient testosterone, delayed or incomplete puberty, breast enlargement, broad hips, reduced facial and body hair, and infertility. So often this doesn't get picked up until puberty because that's when some of these characteristics would really come into play. Other variants, when there's extra copies of that X, so XXXY or XXXXY, have more severe signs and symptoms, things like intellectual disability, more distinctive facial features, skeletal abnormalities, and poor coordination. Men that are diagnosed with Klinefelter have higher risk of breast cancer and lupus. And if you look at the rates, they actually have similar rates of these diagnoses to females in the population. Cognitively, which is my area of interest, they also have delayed speech and language development, and learning disability is really common. So Turner syndrome is females that have 1 X chromosome missing or 1 X chromosome that is structurally altered. Mosaicism can occur when only some of the cells have a chromosomal change. And for the individuals that have mosaic,